Hello everyone and welcome to the lymphatic system. As you can see, I'm not going to be on screen. I know you're distraught. I'm not feeling the greatest, so no me, but at least you get to hear me and that's exciting, right? So we are jumping into the lymphatic system and this is also going to be in accompaniment to the immune system. And you maybe have heard the immune system or, that term before, but let's try to understand it. Alrighty, so jumping into just three functions. The first is going to be fluid balance, and that fluid that we're going to be talking about is going to be called lymph. Lymph, if you want to take a peek at this picture over here, is kind of an accompaniment to blood, and we'll talk about that a little bit more. What this picture shows, though, is a drain put in place after a surgery of some kind. So that area is going to leak various fluids. One of them is going to be blood, and the other one is going to be lymphatic fluid, and we don't want that lymph to be sitting around in those spaces, and we'll kind of find out why here in the next couple of slides. Another function is going to be fat absorption, and when you think of fat absorption as far as the lymphatic system goes, I want you to immediately think of the digestive system. The digestive system is going to be where we are having that fat absorption. The next term underneath fat absorption is lacteals. What I want you to write down is that these are specialized capillaries which will absorb fat. So lacteals are specialized capillaries which are going to absorb fat. And then we have this term chyle. It is pronounced chyle. That is going to be a combination of fat and lymphatic fluid. And then the last function of the lymphatic system is going to be defense. And I want you to kind of write down next to defense is your immune system. So what do we include when we talk about the lymphatic system? We are going to talk about lymph, which, remember what I said on the last slide, that is going to be the fluid that we are discussing in this area. You have lymphatic vessels, which are going to transport lymph. You have lymphatic nodules and nodes, and those are two different things, and we will talk about that. There are a few other structures that are going to be included in the lymphatic system, and they include your tonsils, your spleen, and your thymus. And all of these structures are labeled out over on this diagram over here, which everything is lovely in green. I always like that the lymphatic system is almost exclusively labeled out in green in almost anything that you ever look up. It's kind of interesting. So let's talk about lymphatic capillaries. One of the main things that I want you to get from this section is what lymph is, because it's not the most well-known topic when you are talking about the cardiovascular system. So, and I know that I'm talking about two different systems here, but stick with me. So this first little line here, 15% of the liquid that passes through the capillaries remains in the surrounding tissue. That means as blood is passing through your capillaries, it is kind of dropping off fluid and various other things into the surrounding tissue. This is not a good thing if it stays there. So the lymphatic capillaries are going to pick up that fluid, that lymph, that is left behind from what's in your capillaries. So it's kind of a perfect little kind of recycling system that is going to pick up what is left over, and it's going to carry it to the various lymphatic vessels that it needs to go to. So if we didn't have this system in order to pick up that lymphatic fluid, your tissues in those areas would swell and it would compromise your organ functions. You can see various problems when it comes to lymph not being taken away. It can cause your limbs to swell. It can cause various problems with your organs. Like I said, organs can be compromised. It's a very important system that is going to help you greatly. So here we have a lymphatic nodule, not a node. Make sure you make a note of that. These are nodules. Lymphatic nodules, one of the examples I have for you is called the mucosa-associated lymph tissue, and this is specifically in the digestive system. We also have something called a Peyer's patch, which is a cluster of nodules within the intestine and the appendix. So make sure you know that this is a nodule. One of the things that is a big difference between a nodule and a node is that lymphatic nodules are going to have no capsule. And what that means is that nodes will. So, jumping into your tonsils, we have these various tonsils within the back of your throat, kind of to the top and sides, and kind of just behind your tongue. So we have the pharyngeal tonsils, which are going to be up here if we remove the uvula, so that part of that soft palate. If we remove that, we would see this tonsil up here. You have two palatine tonsils, which are going to be to the sides of your mouth and throat, and then the lingual tonsil is going to be at the very back of the tongue. You can get all three tonsils removed if that is something that needs to happen, possibly an infection or the continuation of various infections can cause you to need to get your tonsils removed. If you have gotten your tonsils removed, it doesn't mean that you have any less of an ability to maintain homeostasis. You probably have more so because for whatever reason those tonsils were not doing any good for you. What we can see in this picture right here is very swollen tonsils. These are incredibly swollen palatine tonsils. They are also infected with what looks like maybe bacteria or yeast. Something that can happen in your tonsils is tonsil crypts becoming filled with tonsil stones. Tonsil crypts are kind of just like a pore. So it's just this little structure kind of coming down here. And in that crypt, you can get these tonsilliths, or tonsil stones. So what happens is this little structure ends up blocking this area and causing an infection. What that can ultimately cause is bad breath. I know I've had a few students in the past who said that they would get these pretty frequently and would have to scrape them out and could literally feel them in their tonsils, and when they would take their fingernail, it felt like a little stone. So kind of gross, kind of cool. All right, remember I said the difference between nodules and nodes is going to be the presence or absence of a capsule. For lymph nodes, we do have a capsule. So make sure that you know that that is one of the big differences between lymph nodes and lymphatic nodules. So lymph nodes are going to be where lymph, remember that fluid, is filtered and will stimulate nearby lymph nodes to process that lymphatic fluid. So in an average person, you have roughly 450 lymph nodes at any given point in time. You have three superficial groups of lymph nodes. I do want you to know their location and in general kind of where they are. So there are three of them. You have lymph nodes in your armpit, your neck, and your groin. So these are three superficial groups that are very important. These areas can become swollen in various times. It kind of just depends, but a lot of the time when doctors are feeling around in your neck, they're feeling for your tonsils, they're feeling your lymph nodes, because remember, the lymph nodes are going to be part of your immune system. So if you are having an immune response, this is going to cause your lymph nodes to kind of go into, in essence, a little bit of an overdrive. Next up, we have the spleen. The spleen is where we are going to have phagocytosis of red blood cells, if you remember from the previous section when we were talking about blood cells and how they are recycled. Purposes of the spleen include immune functions, removal of macrophages, and defective blood cells, again going back to recycling red blood cells. We are also going to have the storage of platelets and blood cells, and there is a difference in the various composition of the spleen, and that is between white pulp and red pulp. Now, I do want you to know the difference, and it's kind of weird, but these dark purple spots are going to be the white pulp. Everything else all around is going to be your red pulp. And this is specifically where we find the specialized lymphatic tissue. So talking more about these pulps, because again, this is where we are going to be talking about this system and what is included. When it comes to white pulp, it is going to be areas composed mostly of lymphocytes that are suspended on reticular fibers. They surround central arteries and appear to form islands in a sea of red pulp. Again, those are the big dark purple splotches. And this is going to be where we have the immune functions of the spleen. The remaining pulp, the red pulp, is going to be where we have the disposal of worn out red blood cells and blood-borne pathogens. So that's all the other functions that we see when it comes to the spleen. Jumping into the thymus. The thymus is going to be located kind of in the very middle of your chest, kind of right above your heart. Your thymus is going to be largest when you are young and smallest as you get older. It is composed of lobes, and honestly, it looks so much like the lung. It's kind of cool. But it is going to help with hormone releasing. We've talked about the thymus before when we talked about the endocrine system, and those are the hormones that it does release. But what I want you kind of to take from this is that these hormones in particular are going to help with something called immune, should be an in, competence. So make sure you know that this is involved in immune competence, and then the remaining hormone that we are going to be kind of applying is that we are going to stimulate differentiation of T cells and the immune response. So just kind of make a note of that for now, and we will get to that a little bit later. All right, so here we have a nice illustration of the immunity. So you do have non-specific resistance, which is going to be over on this side, which is called innate or non-specific immunity. And then you have adaptive or specific immunity. And what I want you to know with that is that it is specific immune cells and proteins. So this is going to include specific immune cells and proteins. Right, and I am going to draw out a little diagram here. I'm just going to pause my audio for a second and draw that in for you guys, so just give me a moment. Now I realize that I probably only stopped talking for about a second when it comes to this video, but I was working on drawing out this graph that I want you guys to write down. And this is going to be what I really want you to know between your innate immunity and your adaptive immunity. And the first thing is the response time. Your innate immunity is going to take just a few hours basically to respond to whatever it is it's responding to, whether that is an infection of some kind or some type of pathogen, who knows. Your adaptive immunity is going to take days to respond. Your innate immunity, the specificity of it is going to be limited and fixed, while your adaptive immunity is going to be highly diverse. In regards to how each immunity is going to handle a repeat infection, your innate immunity is going to be identical to your first infection. Meanwhile, your adaptive immunity is going to be very, very quick. It is going to be able to respond much faster than it did the first time. Kind of like it learned from the first time, it's able to respond better. The major components involved in your innate immunity are going to be barriers and phagocytes. And with the adaptive immunity, that is going to include lymphocytes, antibodies, T cells, and B cells. So if you need to, I would go ahead and pause this slide. We are going to start moving into these various components of the innate immunity. We will talk about each one briefly, but if you need to pause this slide to write down this little graph, I would suggest doing that now. All right, so the first, you probably just heard my cat sneeze, so cute. The first of our non-specific resistance that we're going to talk about is going to be physical barriers. What I want you to think of with physical barriers is your skin and mucous membranes. And then of course you also have various fluids that are going to help also act as a barrier or a deterrent towards pathogens, whether they be bacteria or viruses or whatever might be on the outside of your body that wants to get in. So this includes tears, saliva, and urine. They are all going to have specific chemicals that are going to help fight off invaders. There is a term I do want you to know that is going to be involved with tears, saliva, and urine. And I'll write that out here. So lysosomes, and then write down what I'm about to say. So lysosomes are going to destroy bacteria. They are made in all types of secretion, such as the ones mentioned above, so tears, saliva, and urine. So this is also going to be found on your skin as well in some capacity, and that is just to kind of fight off that bacteria. So make sure you know that we are specifically talking about bacteria. Moving on to phagocytes. So what I want you to really get from this slide more than anything is that phagocytes are going to engulf whatever it is they are trying to get rid of. And there are a few different steps with phagocytosis, but honestly, I'm not going to ask you about any of it, so don't worry about it. I do want you to know microphages versus macrophages. Macrophages are going to include a few friends that we've talked about before, neutrophils and eosinophils, so those various types of white blood cells. And if you need to go back to the cardiovascular system when we talked about blood to know what they do, please refresh your memory on that. And then macrophages. There are two different types of macrophages, fixed and free. Do you know the difference? Fixed is going to be permanent residents of various organs and tissues, while free are going to travel throughout the body and specifically go to areas that need them. So make sure you know microphages and macrophages. Again, the only difference is size, so being big or small, and then kind of what they do here. Now we are moving into some of the kind of fun stuff, in my opinion. I think the immune system is really quite fascinating, and there's various cells that go along with it that I think are really, really cool. So the first that we're going to talk about specifically when we are talking about immune surveillance is going to be NK cells, which is going to be actually natural killer cells, which is probably one of the coolest names for anything in the body, in my opinion. What these are going to use for detection is antigens, and we've talked about antigens before. Remember, antigens are going to be those specific little studs on various cells that let you know that they're supposed to be there or that they're not supposed to be there. If they don't have the right antigens, then they aren't allowed to be there, and your immune system is going to respond in that capacity. So there are these nice little steps. Honestly, as long as you know that we are using antigens for detection, and specifically it's like having the right ID to get into maybe a bar or a club or something, as long as you have the right ID, you can stay. If you don't have the right ID, if you have what this is talking about, a foreign antigen, you are going to be attacked, and the goal is to kick you out. So kind of important. And while we are on this slide talking about natural killer cells, I do want you to write down these various things here that I am going to type out for you. All right, so I do want you to write down these three cells up here. Make sure you know them kind of in relation to as they are. Also, I have a little bit of a way to kind of remember what they do and kind of their importance. So natural killer cells, or NK cells, if you want to think of it in a way that you can kind of understand a little bit better than just how it's written up here, is think of NK cells as like police officers. Police officers are going to kind of patrol for any crimes, and literally that means like anything, right? So they are going to be the first responders. They are also going to be the individuals that are going to find the problem and kind of bust anybody in that situation if it's not a favorable one, right? So they are going to go after kind of any criminal, right? B cells, on the other hand, which I have put over to the side, that these other two are going to be more specified. B cells are going to be like the FBI. The FBI is going to not really care about lower level criminals. They are going to go for somewhere kind of in the middle or a little bit higher up than just a low level criminal, right? Kind of like murders, if you want to think about it that way. So they're not going to worry about smaller offenses. They're going to go after kind of the heavy hitters. While T cells are going to be more like the CIA. And specifically the CIA is going to deal with specialized for bigger terrorists. So these are going to be the cells that are going to go after the really, really, really bad infections. And a lot of the time, all of them have a tendency to work together, but in the end, they all have a little bit more of a specific function, and this is the way I like to remember it. Moving on into interferons, these are going to be small proteins that are going to protect against specifically viral infections. And we do have these various types of interferons. that I do want you to know, and you can literally pause the slide and read through them. As much as on the slide is as much as I want you to know. Next up, we have the complement system, which is going to be where the name is referring to the fact that the system complements the action of the antibodies. And you have over 30 special proteins from the system working for you. These proteins are going to interact with one another in chain reactions or cascades. If you remember talking about hormone cascades, these are very similar. One protein is going to interact with another, and that might interact with another one, and so on and so forth until whatever it needs to get done gets done. One thing I will point out on the slide, don't worry about these two possible pathways. This is going to be a part of your non-specific resistance that's going to be a little bit more relatable, I think, than anything else. This is going to be inflammation and fever. The purpose of inflammation, make sure you write this down, is to get more blood to the area. So what you're going to experience is going to be local swelling, redness, heat, and pain. And most of that is going to typically be from the blood being pulled into that area. The effects of this inflammation usually is going to be your injury or whatever it may be being temporarily repaired, and your defenses will be mobilized. So if you've ever gotten, I don't know, a splinter, let's say a splinter, what happens with a splinter is the area around whatever it is that is sticking into you, whether it's a little piece of wood or glass or metal, it's going to puff up and it's going to hurt, right? If you have a big enough cut or wound or something, usually you can feel some heat coming off of it and it'll become red and swollen. So if you need to think about kind of inflammation, think about a splinter or maybe a paper cut or something. Next, we have the fever. This is where your body temperature is greater than 99 degrees Fahrenheit. Some people run a little colder, so that's not always a reliable temperature to go with, but it's a pretty average temperature that means you have a fever. What happens when you have a fever is something called pyrogens are going to be circulation proteins that are going to raise your body temp. And pyrogens can be released by pathogens, so something that shouldn't be in your body, or may be released by your own body in order to kill off various pathogens. It sort of depends. Also, fevers can be a result of your immune system naturally fighting off a pathogen. It just sort of depends. Okay, so everything we've talked about up till now, since we had that graphic with the chart, has been having to do with non-specific resistance. Now we're talking about specific immunity, or adaptive immunity, or the other immunity, if you will. So specific resistance is going to attack individual antigens from viruses, bacteria, toxins, or foreign tissues. What you can kind of write down for foreign tissues is going to be like if you get some type of transfusion or a new organ or I don't know, a skin graft or something like that. Not always that tissue is going to be accepted into your body, so that is going to be foreign tissues. Specifically, we are going to talk about now is T cells. Remember, we mentioned them before. That technically is not a part of non-specific resistance. It is specific. So we do have T cells that are going to be part of this specific immunity. And T cells are going to direct attacks on abnormal cells and pathogens, and B cells are going to attack with antibodies. I do want you to write down a few things here. B cells are going to work by identifying specific antigens on the surface of cells. because every cell is going to have an antigen. Every single cell, no matter what it is, an amoeba, a virus, a bacteria, a water bear, whatever it might be, they have antigens on their cells or on the cell. And if it doesn't match what needs to be there, it can't stay. So T and B cells are going to look to see if your cells contain your antigens. So it's double checking that the cells that are there are supposed to be there. And I know I kind of keep repeating myself, but that is kind of the take-home message that I want you to get from this. So if it's a bacteria, it's obviously not going to have your antigens. That means it's not going to stay. So there are various types of adaptive immunity, various parts of immunity itself. I do want you to write down these various definitions. So you have innate immunity. This is going to be genetically determined. I want you to write down that it's like a blood type. It is already programmed into you. It's there. Acquired immunity is going to be what immunity you get when you become exposed to a particular pathogen. So as things happen, let's say like you get the flu, the next time you have the flu, chances are your body fights it off a lot quicker than the first time you had it. Or you don't even notice that you got the flu again because your body is able to respond to it that quickly. There are two types of acquired immunity. Natural, this is going to develop after birth. And induced, which is going to be caused by immunization or vaccination. So chances are you've probably heard in some capacity that vaccines are going to carry a part of a virus within it, so parts of like a dead virus, which is kind of true, not completely true. So without going into it excessively, just know, probably for your own benefit, that yes, there are parts of the virus, but it's not like they're injecting you with the virus. The part of the virus that they are injecting you with when it comes to vaccinations is going to be the part that is going to help you build an immunity to that particular virus. So they are injecting part of the virus into you. It's not active and it's not going to hurt you. It's not like you're being injected with a live virus. It is going to be the part of the virus that is going to help your body build an immune system reaction to that virus, so that if you do become exposed to the virus, your body can take care of it without you suffering from the virus. Next type of immunity I want you to know is passive. There are two types of passive immunity. So natural, which is going to come from your mother's antibodies, typically while you are being breastfed, and induced. So this is administered antibodies after the infection. An example of this would be like rabies. Rabies is going to be a virus that if you are bit or somehow infected by an animal that has rabies, you are going to be administered antibodies after the fact. So as opposed to induced acquired immunity, induced passive immunity is going to be after, induced acquired is before. All right, so moving on into specific immune cells. So we have lymphocytes, and these should be terms that we have seen before in this, this lecture. So we have T cells, B cells, and NK cells. These are all lymphocytes. Within T cells, we have various types of T cells. Cytotoxic T cells, helper T cells, regulatory or helper or suppressor T cells, and then memory T cells. What I want you to make a note of is that T cells and B cells are going to be created within bone marrow. And we've talked about this term before. Immunocompetent. I told you we'd get back to it. Well, here it is. This is just referring to competent immune system cells. And that is going to refer to all of these guys. Alrighty, so moving on to triggering adaptive immunity. The first thing we need to talk about is the fact that antigens are either going to infect cells or are going to be processed by phagocytes. And that process of being processed is going to be called antigenic presentation. This is going to be where we have the antigens of the cells that we don't want to be there presented on the plasma membrane of another cell that is then going to present it to other cells to trigger an immune response. And then it's okay to kind of ignore this for just a moment. We'll talk about that later. But how we do that antigenic presentation is going to be using these major histocompatibility complex MHC proteins. So, sorry, I was writing and talking and it's almost impossible for me. So that is the how. We are going to do that processing by phagocytes. So these major histocompatibility complex or MHC proteins are going to be genetically determined membrane glycoproteins. So that just means that they are proteins plus a carbohydrate. Not that that's super important, just know that it's glycoproteins. When it comes to these structures, we do have two types, and we will talk about those next. But really quick, we do want to make sure that we know that the placement of the antigen glycoprotein combination on the plasma membrane is antigen presentation. So that is just as it is putting together the outside of it, it is presenting to other cells. Specifically, it is capable of activating T cells, that's pretty important, by how that combination is presented on the plasma membrane. That brings us to the two classes of MHC proteins. The first class can be found on every membrane that is on every nucleated cell. These proteins are going to act like a red flag. What you can think of when we think about class 1 is flagging viral and precancerous cells. Now I did this little illustration up here to kind of show you what this kind of looks like. So you have one of your own cells, and then you have these antigens on the plasma membrane, so kind of surrounding it, studding it. We've kind of seen this when we looked at red blood cells, right? So what these little antigens are kind of doing is they are making class 1 proteins. So that means that they are stating, hey, I'm meant to be here, I am friendly. It's a good thing. We want that, especially in your own body. If this cell is infected, it's going to have antigens on it that are not good and not yours, and that is not a good thing. So this means that that is bad. We don't want that. It's going to make the wrong protein if it is infected with something, and that is going to tell the other cells that, hey, you're not supposed to be here, not good, no bueno. And if you think about it like this really makes sense, because when we're talking about the class 1 MHC proteins, we're talking about viruses and, I don't even know that I spelled that right, viruses, and we are also talking about precancerous cells. And what these two types of cells are going to do is infect our cells that are supposed to be there, right? They are going to cause some form of infection. You're going to produce these wrong antigens, these wrong proteins, and that is not a good thing. Moving on to class 2, these are going to be found only on membranes of antigen-presenting cells. So these antigen-presenting cells, I want you to know, are called APC cells, and these are going to be phagocytes. Remember, phagocytes engulf whatever it is they're trying to get rid of. And also, these also include lymphocytes, but do kind of keep in mind these antigen-presenting cells. So these proteins are going to tell your immune system that something is different, something is off, something is not right. The APCs will engulf foreign invaders and then present the antigenic fragments on their class 2 MHC proteins. So down here you have this little graphic of an APC which has the correct antigens studded around it, and it is going to engulf this bacteria which does not have the proper proteins on its plasma membrane. Once it is engulfed over here, it is then going to take, the APC is going to take those antigens and present it on its own plasma membrane in order to basically tell other cells that, hey, we have an infection. If you see anybody with this particular antigen, take them out. They're not supposed to be here. So here we have pretty much just exactly what I was showing you on that last slide, just kind of a better graphic here. We have the antigen of whatever it is we don't want to be there. Maybe it's a bacteria, something like that. It's getting engulfed by our macrophage, our phagocyte, or APC, and from there we are going to break apart the antigen and then present it on the outside on our class 2 MHC proteins. From there we are going to talk to, if you will, a T cell and say, hey, if you see anybody with this particular antigen, we don't want them in the body, get them out. So what happens after our macrophage or APC cell presents that antigen to a T cell? Well, it becomes activated and it will start to try to detect other cells that have that specific protein on it. So just kind of going through this slide, if MHC proteins contain antigens that the T cell is programmed to detect, the T cell will bind to that cell, which is called antigen recognition. Make sure you know that. The T cell will have proteins on its membrane that will respond to the MHC proteins. So it is responding to the information that the APC cell has now given it, and it is going to use CD markers, which are called cluster differentiation markers, but it's okay, you can just know it as CD. The T cells will then seek out and destroy abnormal and infected cells that match essentially like the description that it was given by the APC cell. When T cells encounter foreign invaders, they will divide and produce more of themselves. They will also create a memory T cell, which will stay around in the body waiting for another invasion. Now, I didn't talk about them too much before, but we did mention memory T cells when we talked about the different types of T cells. Memory T cells, I kind of like to think of them as little cells that carry around like a scrapbook or a yearbook, and they can go back to when they had that previous infection and say, okay, how did we handle this? Okay, this is how we handled it. Here, there we go. That is why you can have such a fast response to an illness that you've already encountered. This is going to make it to where you can fight off that infection much quicker than the first time. That's why your response time is a lot faster. And then from there, we're going to have our regulatory T cells, which are going to secrete chemical messengers that are going to suppress T cells and B cells, and that will happen after we have fought off that infection. Next up is T cell and B cell activation. Really quick, what I want to kind of get across is sort of the purpose and what our T cell versus B cells are doing. What I want to get across is that B cells are going to help T cells, but not actually kill anything. T cells are going to do the actual killing, and we'll kind of talk about how that happens here in just a second. So, moving into how we start this whole process. We have helper cells are going to go to the area and secrete chemical messengers that are going to stimulate three things. First is the maturation of T cells. You don't naturally have a bunch of T cells running around. They kind of need to be created in order to fight off an infection because they are going to be specific to that infection, right? They're going to have those specific markers that say, hey, you're not supposed to be here, I need to get rid of you. So we have to create those. We are going to attract macrophages, so things to engulf our nasty whatever it is that we're trying to get rid of. And then we're also going to attract NK cells as well. From there, we also want to promote B cell activation because our B cells serve a nice little purpose, again, in helping the whole process. And this is how. B cells are going to produce antibodies that are going to bind to specific antigens. What's going to happen with those antibodies is that they are going to punch holes into the plasma membrane of whatever it is that they are attacking. This is going to help the T cells later be able to kill that cell. So when a B cell antibody binds to foreign antigens, this is called sensitization. Once sensitization happens, the B cell is going to undergo activation, but needs an okay from a helper T cell. The helper T cell is then going to bind to the MHC protein and promote B cell activation. This is going to divide into the plasma cells, which is going to create more antibodies, and then also divide into memory B cells. Because again, anytime you see like a little memory B cell or a memory T cell, these are cuties that are going to go around with their scrapbook or their yearbook and say, hey, remember that time that we fought off that one bacteria or that one thing? Well, this is how we did it, and we can do it again. So talking about antibodies. Antibodies are also called gamma globulins. It's a type of protein found in plasma. We can also call them immunoglobulins. Globulins. I did the exact thing I said I wouldn't do. It's not goblins, it's globulins. And globulins are just going to be involved in your immunity in general. There are 5 general classes of immunoglobulins. I don't need you to know them, just know that there's 5 of them. So moving on to antibodies. We have 2 responses that our antibodies are going to undergo. We have a primary response, which is going to be antibody-mediated response to initial antigen exposure, and this is going to take time to develop. You have to have the appropriate B cells to be activated and then differentiate into antibody-secreting plasma cells. The secondary response is going to be triggered when antigens encounter whatever it is that they encountered the first time, right? More extensive process, and it will last longer than the primary response. The results of immediate responses are going to be by memory B cells for specific antigens, and it will appear even if the second exposure is years after the first. And this is where I've been talking about those little adorable cells that I imagine with their little scrapbooks. But memory cells can survive for about 20 years or more, sometimes your whole life. It just sort of depends on what it is. But typically, if you had chickenpox, you won't have chickenpox again. One, chickenpox is a virus and it stays in your system, but you have also developed these antibodies to fight it off when it flares up. Now, if it does flare up again, specifically with chickenpox, as an adult, it is called shingles, and that can be extremely dangerous. So there's not always an exact perfection, if you will, to this process. You can still get sick again, and it could be a lot worse, but for the most part, your body is going to regulate and take care of you in this process, especially because of these memory cells. All right, and that's kind of the end of this first little section. I did delete quite a few slides, so if you have the notes already and you're not seeing that, that means you don't have to worry about that information. You can look through it if you're really interested, but otherwise we are just going to move on to the next topic. So we've talked about how our body handles infections and kind of the process and physiology of that. Let's talk about those specific things that our body attacks. And we do know that they are incredibly small, microscopic in fact. So how can something so small cause so many problems? Specifically, what is hurting you is called a microbe. Microbes are going to be microscopic organisms such as bacteria, viruses, algae, fungi, which include yeast, and protists. Protists you can think of as little amoebas or little organisms like rotifers that live in ponds, or if you're familiar with water bears, or I think they're also called moss piglets. They're really cute, but take a peek at those if you need a picture of them. They're adorable. But that is what it includes. And not all microbes are harmful to your body. In fact, there are some microbes that we really enjoy. When it comes to cheese, yogurt, bread, beer, certain medications as well, these are usually produced by bacteria, which are really important if you really like those things, right? And you are going to find microbes on literally every surface you've ever come into contact with. They literally exist everywhere. There's no way to not interact with them in some capacity. It's just when they become too much, or it's a certain type that becomes too much, that's when we start having problems. One of the biggest purposes that we have of various microbes is for decomposition. I want you to think of like how bodies decompose. This is going to be in part thanks to microbes. So they are very, very important. When microbes are quote unquote bad, or they have disease-causing agents, that's what we call pathogens. So that is only when they are not something we are welcoming into our existence. It's something that is going to cause you to become sick in some capacity. Starting with bacteria, do make sure you know that bacteria is going to be a single-celled organism. So a single bacterium is going to be single-celled. It has a cell wall surrounded by a capsule. Usually they have a flagella, which is going to cause motility. You can kind of think of how a sperm looks. That is also a flagella, that little whip-like tail. Some bacteria are going to contain something called a pilus. These are going to be elongated hollow appendages that transfer DNA, either from one bacteria to another or from a bacteria to another cell. These can reproduce rapidly in favorable conditions, such as warm, wet environments, especially if there is some type of food source like a sugar or something like that, or lipids. They're going to go wild. And they can produce toxins. These are going to be molecules that inhibit cellular metabolism. They can also become a toxin themselves. And we'll kind of talk about a few things that bacteria can cause here in just a bit. Moving on to viruses. Viruses are one of those special organisms that it's sort of hard to tell if it's alive or not a living organism. Not necessarily that it's alive or dead, but it is contested whether or not they should be classified as living organisms. And if you want to kind of like check out that debate, go back to the very first lecture, I guess, because I do talk about it there. So, what viruses need to survive is going to be a host or host cells. Not unlike a pirate, they are going to invade host cells, take over. Once they're inside, they're going to kind of wreck everything, and in the virus's case, it's going to replicate inside the host cell, essentially destroying the host cells, and then leave and find another host cell to invade. Something that should be noted about viruses is that they are acellular, which means that they are not composed of cells, and viruses are substantially smaller than bacteria. And then over to the right, we have this really, nice and visual graphic of various viruses that we know of. A lot of people think of bacteriophages more than anything when they think of a virus, but now, due to the popularity of information that we have on COVID-19, we see that a lot of viruses actually look like HIV, hepatitis B, and influenza. Ebola is kind of one of those really weird viruses that they kind of just look like a little worm. But typically this is going to be how we have viruses. So some infectious diseases and human health. A few terms that I want you to know. Epidemic. This is going to be more cases of a disease than expected in a certain area for a certain period of time. An outbreak is classified as the epidemic being confined to a local area, and a pandemic is going to be a global epidemic. So, for example, COVID-19 is a pandemic in this instance. And then the term infectious diseases is just diseases that can spread, essentially, and these are going to be monitored by the CDC and WHO. So where do diseases even come from? Because they don't just pop out of nowhere, and we are getting new viruses every once in a while. So how is that happening? So the term emerging diseases is going to be infections that have recently appeared within a population of those whose incidence or geographic range is rapidly increasing or threatens to increase in the near future. So viruses that are new or increase in kind of popularity, if you will, are usually caused by an exposure of humans to animals or insects. So when you think of animals, you can think of, say, civets or bats or something like that in maybe marketplaces or something. We'll kind of talk about that here in just a minute. Or insects. I want you to kind of think of mosquitoes carrying malaria. The other thing to make note of is that viruses and bacteria mutate or evolve over time. This is why you have to get a flu shot every year, even though you say, well, Rachel, I've gotten a flu shot every year. Why do I have to keep getting them if I have, that little piece of the virus in my system, which is helping me form an immune system or immune response to that virus? Why do I have to keep getting it? Well, glad you asked, random student. The virus itself, the influenza virus, is going to evolve and adapt and mutate over time, and honestly, it is one of the fastest evolving viruses, and that's why you have to get a new shot every year. Now, what the flu shot even is what scientists predict to be the dominant strain or the more popular strain of the flu for that season. So if there's a particular strain, and again, they keep evolving and mutating, they form different strains. And that has become one of the bigger fears of COVID-19, is that it is going to evolve into something else, which may cause more problems in the near future. So a few diseases and infections that we are going to be talking about, the first of which is going to be HIV/AIDS. So what these terms mean is acquired immunodeficiency syndrome and human immunodeficiency virus. So they are still both viruses. One is just the kind of continuation of the other, and we'll talk about that in just a second. So the possible origin of HIV/AIDS is from non-human primates, which had become mutated after infected meat had been consumed. Possibly chimpanzee or gorilla meat, maybe bonobo meat, something like that had occurred. And because there's such a close relationship between humans and primates, it wasn't too much of a jump for this virus to go from that original host to humans. What HIV/AIDS does is it's going to infect and destroy cells of the immune system, specifically helper T cells and macrophages. So that means that those particular cells, which are going to be, as you know, very important to your immune system and your ability to respond to pathogens, that's going to completely go out the window because of HIV/AIDS. Those cells are going to be completely destroyed. That means that a cold can become deadly to someone who is infected with HIV/AIDS. What this particular virus is called an opportunistic infection. It occurs only because the immune system is severely weakened. Again, a cold can become deadly. How HIV is transferred from one person to another, and some prevention of it. So unprotected sexual contact is typically how there is going to be a transmission of this virus. Vaginal, rectal, and or oral genital contact can spread the virus. Needle sharing can also spread the virus. Transfusion of blood or blood clotting factors. So you get injected with infected blood, that will give you the virus. Infants with infected mothers. So just because the mother has HIV does not necessarily mean that the infant will 100% be born with HIV. Before or during birth, there could be the transmission of the virus, but more common is through breastfeeding. That's actually a pretty common way for HIV to be spread from mother to infant. A little misconception globally, the main mode of transmission for HIV is through heterosexual sex. A lot of the time, HIV has been considered a homosexual virus, which is a very dangerous misconception because anybody can have it. So it is always important to wear condoms when giving or receiving sex, making sure to talk with your partner about the last time that they were tested, the partners that they have had, and if they were using protection with those partners. So just be smart, have fun, but be smart. That's the most important thing to do to keep yourself nice and healthy. So what can contain the virus? So a few myths is not good, in my opinion, revolving around the transmission of this particular virus. What can contain the virus is blood, semen, vaginal fluids, and breast milk. Things that don't contain the virus: saliva, hugs, hands, toilet seats, doorknobs, dishes, drinking glasses, food, or pets. And I know it seems kind of silly, but I always like to kind of crush myths, especially when it comes to science and anatomy. Next up, we are talking about HIV testing and treatment. When testing for HIV, you are not testing for the actual virus itself. You are testing for the antibodies from the virus. Another important thing to note is it may not show up. These antibodies may not show up immediately in a test. If you just had unprotected sex with an individual, you are concerned about those potential things. Most STDs, STIs are not going to show up immediately. You should wait between 2 and 8 weeks to get tested. I know that seems like a really long time, but it's important to get tested regularly if you are having multiple partners and you are not using protective measures. HIV can turn into AIDS. There is a life cycle to HIV, which we're not going to go into, but if you want to look it up, it's a relatively interesting topic. But we've only recently started really understanding HIV since 1995, and that is not that long ago. For some of you, it might be quite a while. For myself, 1995 doesn't seem like that far away. An important thing to make note of is that there is no cure for AIDS, but there is highly active antiretroviral therapy, try saying that 5 times fast, which is also labeled as the acronym HAART. This is going to stop the virus from replicating to such an extent that it becomes undetectable and harder to transfer. That does not mean that once you are using this therapy that you can have unprotected sex and be fine. It is a way to keep that individual healthy and not die from a cold, as well as to make it harder to transfer in case something should happen, such as a condom breaking or, you know, the condom falling off or something like that. It is to help prevent that transfer. Moving into the next disease, tuberculosis, also known as TB. This is also known as consumption. Consumption comes from the kind of idea back in the day that tuberculosis consumed its victims from the inside. It is actually from a bacteria called Mycobacterium tuberculosis, and it can survive for a few hours outside of the body, which is kind of what makes it a little bit dangerous and highly contagious. It is the #1 cause of death in AIDS patients. There's no way for an immunocompromised person to fight off this infection. It's incredibly deadly in that case. One of the things that I think about when I think about tuberculosis, if you've ever seen the movie Moulin Rouge, the character played by Nicole Kidman suffers throughout the film from tuberculosis. She's always coughing up blood. And that is one of the symptoms, is coughing, specifically coughing up blood and chest pain. And due to that coughing, it's going to cause this kind of spread by airborne droplets. So the incubation period is between 4 and 12 weeks. It does develop quite slowly for a disease. The bacteria is going to reach the lungs, which are then going to be consumed by macrophages. Other white blood cells are going to rush to the area to wall off that original infection. By doing this, the white blood cells are going to create something called tubercles. These tubercles basically create these little hollow spaces, which is going to cause the lungs to bleed. If the immune system fails to control the bacteria within the tubercle will liquefy and form a cavity, and the bacteria will spread to your kidneys, spine, and brain, which then it becomes really difficult to fight off. But it typically will lead to death if that spread occurs, but if you can stop it quick enough, you are pretty much good to go. It is a very manageable disease now. Back in the day, not so much. Our next disease is malaria, and I know we've already talked about malaria in the last section, but I would be remiss not to mention it again. Malaria is known as the world's worst invisible pandemic. I think this is one of those diseases that people kind of underestimate because it's not happening around us in America, but it kills quite a few people. Just in 2012 alone, 627,000 people died due to malaria. I do want you to know this term vector. Vector is a living organism that transfers the pathogen from one host to another. In this case, our vector is a mosquito, or mosquitoes in general. It is going to be a parasite that lives inside of the saliva of mosquitoes. Mosquitoes will then bite a host, which is going to deliver the parasite into the host's blood system. It's going to travel through to the liver, experience asexual reproduction. The babies of those original parasites will then infect more liver cells and red blood cells. From there, the offspring is going to grow inside of the red blood cells and then burst out, causing a immune response. From that immune response, those offspring from the original parasite are then going to find more red blood cells to hide inside, reproduce, and then burst out again. What this causes in people who have malaria is these cycles of every 48 to 72 hours, they're going to go through extreme pain, fatigue, probably fainting, they might grow unconscious, but it's going to coincide with the parasites bursting out of the red blood cells, which will eventually, if it happens enough, cause anemia. And remember, anemia is going to be where you have a lack of red blood cells that can carry oxygen throughout the body. Next up, we have influenza. Influenza yearly kills 36,000 people. The symptoms include runny noses, cough, chills, fever, body aches, and nausea. Keep in mind that this is an infection of the respiratory tract. A lot of people think of the stomach flu as being influenza for some reason. There's a lot of misconceptions when it comes to the terminology, but when you typically talk about the flu, just flu, you're talking about something occurring in your lungs, in your throat, in your respiratory tract. When it comes to the stomach flu, it is called gastroenteritis. What that includes is diarrhea, cramps, nausea, and then a low-grade fever. That is caused by infected food or water. You could catch it from somebody else, but that means you maybe came in contact with that bacteria, maybe from someone not washing their hands or something like that. But do keep in mind that influenza is different than the stomach flu, and when people refer to flu, that's typically going to refer to the respiratory tract. There are two types of influenza that I want you to know, and that is H5N1, which is known as the avian influenza, and H1N1, which is swine flu. And potentially you've heard of both of these. They were both quite deadly when they were more novel viruses. They are still deadly, don't get me wrong, but we do have vaccines for both of them now. H1N1 is typically going to be a strain that you see seasonally. It did start in pigs. The original outbreak was in 2009. This is also the same or similar flu virus that caused the 1918 flu pandemic, if you are familiar with a little bit of history. H5N1, or the avian influenza, is transferred through wild birds, waterfowl, and domestic poultry. So if domestic poultry seems like something that potentially you would have come in contact with, such as chicken or turkeys, then that probably sounds familiar, right? It does not pass easily between humans, but it is possible and can become more of a problem if the virus mutates. Again, going back to viruses are constantly mutating. So does bacteria. That is the big concern, and that is why you need to get a flu virus shot or a flu vaccine every year. Now, because this is the most recent, at least at the time of me making this video, it'll probably still be relevant in later semesters, but currently it is 2020, and this is a relevant topic. So, COVID-19, it is one of the newest viral pandemics starting in the late 2019s and becoming prevalent in 2020. What this translates into is the coronavirus disease 2019. So that is what COVID-19 stands for. It is caused by a novel coronavirus, which is a particular type of virus. So there are like 300 plus types of coronaviruses. This is just one that was able to be transferred to humans. We've never seen this particular virus in humans before. That is why we are having such a hard time sort of fighting it off, if you will. It is highly contagious as well. It is transferred through droplets in the air, not dissimilar to TB. Masks are preventative measures rather than protective. It is to help the wearer contain whatever they might have. If that is this particular virus, then they are keeping it more to themselves. The proper way to wear a mask is to cover one's entire lower face. So from the eyes down to the chin, wearing your mask underneath your nose absolutely does nothing to prevent anything other than when you talk, I suppose. But it can still be transferred even from breathing. The virus can also remain viable on surfaces for hours to days. Disinfecting is going to help as long as the disinfectant sits on a surface or your hands for as long as that particular disinfectant suggests for your hands. You should probably keep disinfectant on your hands for about 30 seconds or more in order to kill any pathogens on your hands. What is assumed, at least at the moment, is that COVID-19 originated in China and was spread from bats, not dissimilar to SARS, which originally came from civets in marketplaces, which civets actually, it is predicted that they got SARS from bats as well. And if you don't know what a civet is, it's kind of a cat-like creature, but these animals are for the purpose of consuming. So there's a picture of a civet up in the upper corner if you want to look at that guy. So let's talk about antibiotic resistance. What this is where a virus or a bacteria becomes resistant to the mode in which we try to get rid of it. So certain viruses can become like disinfectant resistant. So if you ever look at a disinfectant jar, we kill 99.9% of viruses and bacteria on surfaces. Okay, well, you know, obviously the one that you don't kill is maybe the one we should be concerned about. But that's kind of how it happens. What's interesting, in 1943, we found penicillin. Penicillin was a life-saving antibiotic and also medication, right? 4 years after penicillin was introduced, bacteria began to develop a resistance to it. It mutated that fast, which is crazy. Penicillin is still used today. It's still very, very important. And it's kind of interesting. What happens with antibiotics, people don't become antibiotic resistant. It's the bacteria inside of you that is becoming antibiotic resistant. So that is kind of the concern with taking too many antibiotics. One of the scariest infections that you can get, usually in maybe a gym, nursing homes, or hospitals, is MRSA. That is methicillin-resistant Staphylococcus aureus. This is going to cause staph infections, boils, and hair follicle infections. This is a antibiotic resistant bacteria, which normally is okie dokie, but given the right circumstances, let's say you're in the hospital and you get MRSA, well, if you're already in the hospital, chances are an infection is not going to go well for you, right? But it is normally passed from monosymptomatic carriers, so someone who has it but doesn't have any symptoms, through hand contact. So again, kind of think shared equipment in a gym, nursing homes, and hospitals. And that's all I've got for you from this lecture.